Lipid digestion turns out to be a far more complex sequence of events than we've seen for carbohydrates, and we've seen for proteins, and than what we'll see for nucleic acids in a few minutes. And there's a very good reason for that. If we look at proteins, carbohydrates, and nucleic acids, those are all hydrophilic. They dissolve readily in water, so they dissolve readily in the forming chyme. It means that they come into contact relatively easily with the different types of pancreatic enzymes that get released into the lumen of the small intestine. The same is not true for lipids, because lipids, remember, they're hydrophobic. So when we put lipids in an aqueous solution, when we put them in water, they tend to glob up together, just like if you fill a sink full of water and then you put a greasy pan into the sink, you know, all of the fat globules from that greasy pan, they don't just dissolve into the water, they all float up to the top and they congregate into these large fat globules, and that's because they're hydrophobic. This creates a problem, because now I'm going to have fat globules in my chyme. And fat globules are going to have very little surface area compared to their volume. And that's going to slow down tremendously the rate at which I can chemically digest those lipids. So the first order of business that we're going to have to do with lipids is we've got to get these big fat globules divided up into smaller fat globules that are tiny enough that I can then have chemical digestion proceed at a reasonable rate instead of spending 3 weeks trying to digest fats from a hamburger. So let's look at how this process happens first. Okay. Now, we already learned that there is some chemical digestion that can occur, a small amount can occur, due to the activities of lingual lipase. Lingual lipase was made by your salivary glands, and it only became active once it reached the acidic pH of the stomach. This accounts for the digestion of roughly maybe 10% of the fats you consume in your diet. I have a picture of a baby here breastfeeding because I wanted to tell everybody that there is another type of lipase called gastric lipase, which, as the name implies, is manufactured in the stomach. And in babies, it's produced in very high quantities because their diet is pretty much exclusively breast milk. And so we have lots of gastric lipase in addition to the lingual lipase to facilitate the digestion of the very fat high diet they have. When babies are weaned and start eating solid foods, the amount of gastric lipase that they produce drops dramatically. And oftentimes we don't even produce it at all as adults. Some adults still produce a little bit of it. Anyhow, the combined effect of this is that for adults, we only can really digest about 10% of our lipids thanks to the activities of lingual lipase and a little bit from gastric lipase. The remaining 90% of those lipids are then still in the stomach. They're forming these large fat droplets, fat globules, if you will, and the larger they are, the less surface area I have for pancreatic lipase, which is coming down the pike in the small intestine, to be able to interact with those fat globules. So first we've got to do something to correct that situation. Before I even start chemically digesting lipids in the lumen of the small intestine, I need to first undergo a process called emulsification. The idea behind emulsification is we're going to use bile and the bile salts and lecithin that are actually in the bile to take large fat droplets and divide them up physically. We're not breaking chemical bonds here. Okay? We're just making one big fat globule into a bunch of smaller fat globules. These are called emulsification droplets. And I want to emphasize again, this is a type of mechanical digestion because no chemical bonds are being broken. This is mechanical in nature. It's just to increase surface area relative to volume. And how this is done is that bile acids and bile salts that are present in bile have both hydrophilic and hydrophobic regions. And they will kind of surround areas of triglycerides on the outside, and basically that helps to break them apart into smaller pieces. And because the hydrophilic, that's the blue region here, the hydrophilic region of the bile points outward, it helps these emulsification droplets better interact with the surrounding watery environment that they're in. So that's a first step. You've got to emulsify the fats through a mechanical process to get them small enough that we can then finally have enough surface area to volume ratio for pancreatic lipase to begin doing its job. Pancreatic lipase is manufactured in the pancreas. It's secreted into the duodenal lumen. So in the lumen now, we've got these individual emulsification droplets, and they begin to come into contact with pancreatic lipase. That pancreatic lipase is particularly good at taking triglycerides, such as these guys shown here, and breaking them apart in the presence of water into a monoglyceride and two free fatty acids. So you can see that over here. Here's a free fatty acid, here's a free fatty acid, and here's your monoglyceride that's left over from the triglyceride. Okay, now this is all happening inside this emulsification droplet. When I've digested all of those emulsification droplets, get this all out of the way here. When I've digested the triglycerides in those emulsification droplets, at that point, I'm finally ready to start to do some sort of absorption, okay? And by the way, these emulsification droplets, once they contain the digested lipids, we call them micelles. Okay, so an emulsification droplet contains undigested lipids. A micelle contains digested lipids that are just getting ready to be absorbed. So how does that happen? So these individual micelles, here they are, start to bump up against the brush border. And as they do that, individual free fatty acids will be allowed to pass through the plasma membrane of that cell, and individual monoglycerides will be released from the micelle and will diffuse down their concentration gradient and into this intestinal cell. So every time a micelle bumps up against the brush border, a few of these fatty acids and a few monoglycerides are allowed to pass through the plasma membrane, and they are incorporated into the absorptive cell, into that intestinal cell, and then something odd takes place. Then we get the Golgi complex involved. Remember from Bio 181, the Golgi complex is sort of the shipping and packaging center of the cell. This is where we do the final construction assembly of things like glycoproteins, and then we package them up for export out of the cell. Well, in this case, the Golgi complex will grab those two free fatty acids and that monoglyceride, and it will join them back together into a triglyceride again. And then it packages up all these triglycerides into a little package, if you will, called a chylomicron. Okay, and here it is. Here's my Golgi complex. Here it is releasing these chylomicrons full of triglycerides. Those chylomicrons then migrate over to the basal side, that's opposite from the lumen, basal side of these cells, and they are exocytosed. And they release their contents, all these triglycerides, into this extracellular fluid. They diffuse down their concentration gradient and are picked up by the lacteals. Now you might be wondering, why the heck would we go to all the trouble in the previous slide here of having pancreatic lipase break apart triglycerides into a monoglyceride and two free fatty acids if we're only going to just turn around and inside of these intestinal cells we're going to remake the triglyceride? And the answer to that is this. The triglycerides that are out here in the lumen of the small intestine are just too large. They cannot pass through that plasma membrane. It just won't happen. So we've got to break them down into smaller pieces, import those smaller pieces into the intestinal cell, then construct them back together and exocytose them out the other side in the form of these chylomicrons. So these chylomicrons, just so I want to be clear here because I think maybe I wasn't, each of these vesicles here contains, hundreds if not thousands of chylomicrons, and each chylomicron is filled with a bunch of those triglycerides and some cholesterol and some phospholipids. So these little dots here each represent a chylomicron. Each of those chylomicrons has thousands of molecules of lipids within them, and those get exocytosed. You'll be happy to know that the process of digestion for nucleic acids is pretty darn simple comparatively speaking. And that's partially because nucleic acids, like proteins and carbohydrates, are hydrophilic. They are readily dissolvable in water. They easily come into contact with enzymes that are present in the lumen of the small intestine. And that's the first location where they experience chemical digestion. When these nucleic acids come into contact with pancreatic nucleases, this is a class of enzymes that digest RNA and DNA, they begin to break apart. Now, there are specialized types of pancreatic nucleases. We have ribonuclease, as the name implies, it specializes in digesting RNA. And we have deoxyribonuclease that preferentially breaks down DNA. It breaks down ribonucleic acids and deoxyribonucleic acids into individual nucleotides. So, for example, we'd have RNA plus water in the presence of ribonuclease to give us individual nucleotides. Same thing would happen for DNA in the presence of deoxyribonuclease. Now remember from Bio 181, a nucleotide consists of 3 distinct parts: a phosphate group, a sugar group, and a nitrogenous base. A nucleotide is still too big to be absorbed. So once I've gotten individual nucleotides released from the RNA and DNA, I'm not done. I still have to incorporate some other activities, and this time, rather than happening in the lumen of the duodenum, we're going to look at events that are occurring on the brush border. There are some brush border enzymes called nucleosidases and phosphatases that specialize in releasing these individual parts, breaking the phosphate group off from the sugar group, breaking the nitrogenous base off from the sugar group. So as these individual nucleotides bump up against the brush border enzymes will take apart the phosphate group, will take off the nitrogenous base, will take off the sugar group. And now that we've got these individual much smaller molecules, those can be readily absorbed using different membrane carrier proteins. Then we get them into the intestinal cell and eventually out the other side and heading towards the blood. The last thing I want to discuss very briefly is vitamin digestion and absorption. Now, remember that I told you at the very beginning of all of the digestion lectures that vitamins, for the most part, are relatively small molecules. And so chemical digestion really isn't necessary. We do still have to do some mechanical digestion just to get, small enough globs of food that the vitamins can then be readily absorbed. But things like vitamins A, D, E, and K are very hydrophobic. And so they tend to get absorbed in those chylomicrons, first in the micelles, and then they get formed in chylomicrons. They tend to be readily absorbed along with other lipids. By comparison, water-soluble or hydrophilic vitamins like vitamins B and C tend to just be absorbed via diffusion. They move down their concentration gradient and into the intestinal cells and then are introduced in the blood. The exception to that is vitamin B12, and as I told you before, vitamin B12 is an extremely large molecule. It's too large to be absorbed without some help. The help it gets is that intrinsic factor, which is shown here as this kind of Pac-Man shaped guy. An intrinsic factor binds with vitamin B12, and then it facilitates the movement of vitamin B12 into the cell. At that point, the intestinal cell, separates the vitamin B12 from the intrinsic factor and releases the vitamin B12 into the blood, and the intrinsic factor can then be recycled and used again for another round of absorption. That's all I'm going to say about vitamins, and that concludes chemical digestion.